Hello, I am Javier González-Canales, Research Assistant in the Department of Edapology at the Politécnica University of Madrid, and I'm going to talk about the poster called Previous Crop and Nitrogen Fertilization Effects on Soil Health Indicators in Wheat. Soil microbiome plays an essential role in biogeochemical cycles and ecosystem service provision. Intensification of agricultural production has compromised soil health, resulting in the degraded agrosystem we have nowadays. In this context, the entry of legumes into the rotation has been considered a strategy to stimulate symbiosis and improve soil microbiota abundance and activity. The crop residue incorporation in soil means a higher nitrogen input into the soil, in addition to the negative effect in the nitrogen fertilization. Beneficial soil microorganisms, including arbuscular mycorrhizal fungi, can be adversely affected by high nitrogen levels. The nitrogen fertilizer dose reduction applied after legumes can enhance microorganization with potential benefits in crop nutrition. Our aim was to evaluate the effects on previous crop, legume or grass, and nitrogen fertilization on several soil parameters in subsequent wheat crop as a part of a larger experiment. The field trial was carried out in the experimental farm La Canaleja in Alcalá de Henares, Madrid, Spain. We considered two factors: precedent crops, Pisum sativum or Ordeum vulgare, and two nitrogen fertilization levels on wheat in a randomized block factorial design with 4 replication. The chosen crop was a traditional wheat variety Nogal in a rain-fed condition. Tillage was done with a disc harrow and cultivator. We applied fertilization for an optimal level of potassium as phosphorus and a post-emergence herbicide. Soil sampling was carried out in the 0-15 cm deep at the flowering wheat stage. We measured several biological, chemical, and microbiological soil properties as percent of mycorrhizal colonization on wheat roots and hyphal lengths, basal respiration, soil microbial biomass carbon, or total organic carbon. Data analysis was carried out by ANOVA with a mixed linear model for a split block design. Mycorrhizal colonization on wheat was reduced by 31% on fertilized wheat compared to unfertilized wheat. This confirmed the initial hypothesis. The reduction with fertilization was rather after pea than after barley. When nutrients are readily available, plants allocate less photosynthase to root and mycorrhizal symbionts. Extraradicular hyphal length in wheat after barley increased by 27% compared to wheat after pea, regardless of nitrogen dose applied. Difference in root structure and root biomass may explain this difference. A similar trend was shown for basal respiration at 24 hours. Microbial biomass carbon was favored by both preceding pea and nitrogen fertilization. The metabolic coefficient showed lower values in whether after pea than after barley, 32% lower. That supposes more efficient use of carbon by introducing pea in the rotation. The microbial coefficient suggests an improvement after pea, indicating that this treatment favors conditions that establishment of microbial communities an upper degree. Our preliminary results confirms that rotation influences the soil health parameters studied, and in this case, its effect is higher than the effect of nitrogen fertilization. The precedent barley trends to improve mycorrhizal parameters in wheat better than the precedent pea. However, in general, there is a positive effect of pea as a precursor crop for a winter wheat in a Mediterranean environment by providing conditions that reinforce a more efficient use of carbon and a greater stimulation of microbial population.